Assalamualaikum everyone. First of all, I would like to thank all the present committee of ICAZ 2022 for providing my MPhil research work to the Scientific Society platform. My name is Laiba Ameen from the Department of Zoology, Government College University, Faisalabad, and topic of my research work is peanut husk and leaf fumitory extract as antioxidant and antimicrobial agents. First of all, introduction. Natural products have therapeutic potential against various diseases such as inflammations and cancer. Since ancient times, natural products have been utilized to prevent and cure microbial infection. Natural products have provided a major foundation in developing the antibiotic drugs. Peanut husk as a natural product is effective for many diseases. Traditional Chinese medicines is utilized to provide the peanut skins to cure some illness. To cure many illness, leaf fumitory belongs to family Fumicaceae, includes small branching animal herbs, helps to create many diseases. The aims and objective of my study is that to perform phytochemical analysis of Fumiria parviflora and Ehrlichia hypogaea extract. Here, the Fumiria parviflora is the scientific name of the fine leaf fumitory and Ehrlichia hypogaea is the scientific name of peanut husk. To investigate an antioxidant potential of both these plant extract. Third, isolation and characterization of strains to check antimicrobial activity. Fourth, to examine the antibacterial activity against the selected strain. Methodology. In my present study, I have been collected the plant's parts from the Faisalabad. Then I dried it under the shade and grinded it into the powder form, and then have been subjected to the green extraction method. The green extraction then concentrated through rotary evaporator. The prepared extract then used for different analysis. First of all, I have conducted the phytochemical analysis in order to check the presence of alkaloid test, saponins test, phenolic test, flavonoid test, and steroid test. In order to check the antibacterial activity of plants, extracts against the selected strain have been performed through well diffusion method. Radical squenching activity of the plants have been investigated through DPPH assay. The results, if I talk about my results, Fumiria parviflora and Ehrlichia hypogaea extract shows the presence of phytochemicals such as flavonoids, saponins, steroids, tannins, and alkaloids. Both extracts, if we will talk about the antibacterial, then both extracts shows the antibacterial activity as well. But the Fumiria parviflora shows the best or more potent against the Bacillus cereus as compared to the other selected strains. The mean plot representations of the zone of inhibition of Fumiria parviflora against Bacillus cereus has been shown in the diagram or in the graph. And the comparative graph of antibacterial activity of Ehrlichia hypogaea and Fumiria parviflora against Bacillus cereus also shown in the graph. Here, the antibacterial activity of Fumiria parviflora against different strains has been shown in the graph, and the Bacillus cereus shows the highest potency or more potent results as compared to the E. coli, Pseudomonas aeruginosa, S. aureus, Salmonella enteриca, Aromonas hydrophila, and Pasturula multisuda. If we talk about the Ehrlichia hypogaea, that was the peanut husk. The peanut husk shows the more potent and more antibacterial activity as compared to that of the Fumiria parviflora, because the Ehrlichia hypogaea have more ability to kill the bacteria, and the inhibition zone of Ehrlichia hypogaea is more potent against the E. coli bacterial strain. Mean plot representation of the zone of inhibition of Ehrlichia hypogaea against E. coli has been shown in the graph. Comparative graph of the antibacterial activity of Ehrlichia hypogaea and Fumiria parviflora against E. coli has been shown in the graph, and it shows the more potency of the Ehrlichia hypogaea extract as compared to that of the Fumiria parviflora. The antibacterial activity of peanut, that was Ehrlichia hypogaea, husk extract against the different strains has been shown in the graph. The E. coli shows the more potency of antibacterial activity as compared to the other selected strain, that was the Bacillus cereus, Pseudomonas aeruginosa, Salmonella Salmonella enterica, S. aureus, and Aromonas hydrophila, and Pasturula multisuda. Both extracts shows the radical squenching activity, but the peanut husk shows the higher IC50 value, that is 3.490, as compared to that of the IC50 value of, which is near to the IC50 value of the ascorbic acid, that was 3.37 microgram per ml, which shows the Arachis hypogaea have higher radical squenching activity, which is near to the our standard ascorbic acid as compared to that of the Fumaria parviflora. Here, the graph will show the radical squenching activity of peanut husk and the Fumaria. The ascorbic acid shows the blue line, the peanut husk shows the red line, and the Fumaria shows the yellow line. So the blue line more closer to the red line, which shows that the ascorbic acid, which is our standard, have more potency or have a more radical squenching activity, and our peanut husk is more also have the more radical squenching activity as compared to that of the ascorbic acid. Conclusion: based on our results presented above, it was concluded from this study that according to biological assessment, it is concluded that both extract, that is Arachis hypogaea and Fumaria parviflora, are more efficient recording biological activities. A major antimicrobacterial and antioxidant agents, both extract provide us opportunity to learn more about them in the field of herbal medicine. It has been determined that Arachis hypogaea and Fumaria parviflora are the source of medicinally active ingredients and have a variety of pharmacological effects. Therefore, it is exciting to discover a new treatments using these substances. Moreover, it has been discovered that they have a strong antibacterial properties and can be utilized to isolate antimicrobial medicines. These have considerable antioxidant potential as well. These results recommended that peanut husk Arachis hypogaea have good antioxidant properties and can be used instead of ascorbic acid. These results also recommended that there is a need to investigate the biological properties of other fruit peels that are discarded. Thank you for your attention.